Okay, welcome back, everybody. Today we're going to discuss biological beginnings. What does that mean? Well, we're going to discuss all of the biological and physiological correlates and processes that relate to behavior, cognition, and affect. So, what does that mean? Well, that means if there are hormones present within an individual at higher than normal concentrations, when behavior X occurs, maybe we can infer something about the relationship between that hormone, its concentration, and the behavior that was produced. Now, this only works somewhat well because the problem that you run into when you're relating these physiological processes to behavior and mental processes is that their relationship is typically contextual. And so if you weed out the context, if you get rid of the context within which the behavior occurs, you lose the relationship between, or you may very well lose. It might be the case that it's so connected that you don't lose much of it. But typically it's the case that when you weed out the context, when you ignore the context of the behavior, then you lose the relationship between the physiological process, the neuronal activity, and mental processes along with the behavior that occurs. But we're going to talk about these biological correlates today. Okay, evolutionary psychology needs to be discussed at this point. Evolutionary psychologists are going to look at the adaptive purpose of behavior and the adaptive purpose of mental processes. Well, what does that mean? What the hell is adaptive mean? What are we discussing? Well, adaptive means, does it allow this individual to survive in their environment to a point where they can reproduce and pass on their genes? Okay, going right in line with evolutionary theory. And now we're not going to discuss all of the tenets of evolutionary theory. You can get that in a separate course. But we need to talk about evolutionary theory because it so much has contributed to medical science, and it's so much contributed to our understanding of behavior and why behavior exists in the forms that it does, and furthermore, why behavior in our species takes time to develop. For example, why is a giraffe born walking and running, but a human is born immobile, can't even move, can't do anything without its parents? So why do those differences exist? Well, if you live in Africa and there are a lot of very scary animals that are chasing you, trying to eat you, you better be born ready to run. And evolutionary processes have made that possible. Evolutionary processes have also, and our understanding of them, have also allowed us to attack certain types of disease and disorder from various fronts so that we can help aid people in the recovery processes. Evolutionary theory is also going to allow us to look at mental disorders in a new light. A lot of people look at mental disorder as, or historically have looked at mental disorder as, inhibiting or debilitating in some way. And there are features of mental disorders that are that. There's no doubt about it. However, we also want to look at, are there some features of these mental disorders that are adaptive given very unique and chance circumstances? And that seems to be true with at least some mental disorders. All right, so Darwin's biographical sketch of an infant. His goal was to document the emergence of expressions. This was a case study. It was a case study with much anecdotal information by Darwin, but Darwin made some very astute observations, and this was of his own infant. So as you can imagine, there's going to be some bias there. But when you read this article, there's not a whole lot of bias. He tries to really look at this through the lens of a scientific mind. However, there is some bias in there when he starts talking about his particular interactions with the infant. But some of the infant reflexes that he noticed were sucking, blinking, yawning, and screaming. He said that these were under innate controls. We have these when we're born because they're so important. And if we don't have them when we're born, then bad things can happen. For example, if you don't know how to suck when you're born, you're not going to eat very well. And if you don't eat, you're not going to get the nutrition you need, and it's not going to work out so well. Emotional expressions are there for as communication tools to disclose what the individual's feeling. So pleasure was an emotional expression that was shown by Darwin's child when Darwin's child fed. This was information that could be interpreted by the parent to say, yeah, I did what I needed to do. The infant got what they needed. Okay. Affection, smiling usually occurred about 2 months. He said that this was quite early. And the reason that it emerged so early is because smiling is a way to form that social bond, and social bonding is very important in our species. Without it, we don't survive so well. Fear, crying and making noise happened at a few weeks of age. This is another one of those very necessary emotional expressions. If we can't do it very early on, things don't usually turn out so well. If we can't scream when we're unhappy or when we're scared, we don't really know and can't get that feedback from somebody else as to whether or not it's appropriate for us to be scared, or if it's something genuinely scary, then we need help with it. We're only a little tyke at this point, and if we don't have the assistance, we don't get out of harm's way. Anger, he couldn't really determine when this occurred. He felt that there was a lot of social learning with anger, and we know now with Albert Bandura's work that there is much social learning with aggression. Now, aggression can be quite different than anger. You can be aggressive without being angry, but here, what Darwin is alluding to is he's saying that, okay, we learn aggressive, angry responses by watching others. And that's what we know of aggression today. Okay, we learn how to be aggressive and angry by watching the people around us be aggressive and angry. If they're not aggressive and angry when we're growing up, then it's very unlikely that we're going to become aggressive and angry. All right, to continue this, Darwin thought that there were complex associations that were formed at various ages. There was a morality that was formed, and he also chimed in on language development. Associated ideas occur when excitatory stimulus is present at 5 months of age, and the individual starts to relate those stimuli with other features of their environment. He also talked about operant mechanisms and praise as being important for the infant, and the infant can understand this about 7 months. Now, we know that that can actually occur a little bit earlier than 7 months, but Darwin's kind of on the right track here. And we also know that associative learning by stimulus associations occurs prenatally. It can occur much earlier than 5 months, but for Darwin, it was 5 months. That's what he noticed in his infant. Emergence of morality was about 13 months. This was expressed as guilt in Darwin's infant. sharing occurred about 2 years. This is kind of important. Darwin's touching on something that we're going to discuss later called theory of mind. And it seems that he very well documented the emergence of theory of mind in his infant, in that theory of mind does kind of, it can emerge a little earlier, but it's usually there about the age of 2 to some basic form. And a 2-year-old wanting to share is, I mean, that's age appropriate for their use of theory of mind. And productive vocabulary occurred about the age of 1. Now, receptive vocabulary is different. That occurs when you understand what the words mean. Productive vocabulary is when you start asking for things. And in this case, it was for like food. And I'll let you watch those 2 videos. You'll learn a little bit more about emotion as it relates to humans, non-human animals, and then you'll also see a little discussion about genetic differences between species, with a particular emphasis on the genetic differences between chimps and humans. Okay, this is an image of a human cell, and what I want you to know is I want you to know a little bit about the nucleus that contains the genetic information, the DNA, are contained there. And then I also want you to know a little bit about these ribosomes. Ribosomes are going to code for proteins, or sorry, not code for proteins. They're going to produce the proteins from the code that is copied from the DNA. Okay. Then we have mitochondria, and mitochondria are the powerhouses of the cell, and they also contain their own DNA. What's interesting about the mitochondria is you get your mitochondria from your mother. There's some discussion as to whether or not sperm can transfer some of this mitochondria, because they do have a little bit of it, but it doesn't usually make it in to the cell. But in most cases, mitochondria come from mom. And that's a very important thing, because if you start tracking maybe in an animal, you don't know mom, you don't know dad, but you can know mom by looking at mitochondrial DNA in individuals that are around the offspring. And so, I mean, that's helpful. That's helpful to know that information. Okay, genetics. This is the double helix, and DNA contains, the code for life. Okay, that's what's contained within this double helix, and really all it is a code to make proteins. Okay, based on the sequencing of these nucleotides, we will get different proteins. And if you haven't taken genetics, which many of you probably haven't taken a genetics course, I encourage you to take genetics as you move into your upper level courses. There's a lot to learn there, and I really encourage you to take it. Now, 2 processes you need to know is transcription and translation. Transcription is where an enzyme goes in there, and where we go in there. Yeah, we're gonna where an enzyme goes in and it unzips this double helix and it starts copying the code. Okay, that's transcription. Then translation is the process of making that code into something meaningful. So that code that was copied goes to a ribosome. The ribosome then produces a protein from the code. All right, some genetic principles. I want you to know the difference between dominant and recessive genes. Dominant genes are usually expressed, or are going to be expressed, when in the presence of another gene, a recessive gene. When 2 dominant alleles are present, you have the dominant phenotype. When a dominant and a recessive allele are present, you still have that dominant phenotype in most cases. Sometimes you get some odd blends, and you see that down there with the Great Danes. And then usually to get the recessive phenotype, you have to have both recessive alleles there. That's Mendelian genetics. There's a lot that we now know that kind of tweaks these Punnett squares a little bit. So it's kind of interesting. The interaction between the environment matters. Hey, polygenic influences are where we have multiple genes weighing in on the features of this individual. I'll let you watch those two videos to get a closer look. Excuse me. Gene-environment relationships are important. We have passive gene-environment relationships where the environment is pretty much coming from the parents and the offspring is just kind of a passive participant in whatever environment they're placed in. Evocative is where the individual's temperament is going to start shaping its environment, thus leading to other environmental influences on the individual. And an active gene-environment interaction is where the individual is actively selecting the environments within which they want to participate. So the difference between evocative and active is maybe the individual is shy, and so they withdraw from social interactions, and that changes their environment ever so slightly, and they have different life outcomes. An active selection of the environment is the individual is actively selecting tasks that they work on their own that exclude maybe other people. And it can be the inverse as well, can be true. And so you have some epigenetic influences. I'll let you guys think on those epigenetic influences a little bit more. That's nutrition, environment, interactions with the genes. Okay, all of those things can tweak the way that genes are expressed. Okay, Down syndrome is trisomy 21. There are other trisomies where we have 3 chromosomes rather than the 2 that we're supposed to have, and we can get some deleterious effects that can be chronic and pervasive. Here are some of the features of Down syndrome. One thing I want you to know is as mom gets older, the risk of Down syndrome goes up. As dad gets older, the risk of Down syndrome goes up. Okay, and it tends to differentially express itself across different ethnicities. So African American parents are less at risk for having Down syndrome offspring. We're going to see in other cases where different ethnicities are more likely to have other things, for example, other ailments and diseases. For example, the Native American population, because of our westernized diet that they haven't been historically exposed to, has led to a very high prevalence and high rate of type 2 diabetes within their populations because they didn't eat all of these westernized processed foods, and now all of a sudden they're eating westernized processed food and their genetics doesn't know really how to interact with those nutrients. And so they're processed differently. So we're going to see that across different ethnicities have there are just subtle differences in the genes that lead to these different things. Okay. All right, sex-linked chromosomal abnormalities. Klinefelter syndrome, Fragile X syndrome, Turner syndrome, and then the super male syndrome. Okay, not really a super male or a super criminal, not really an overexpressed type for male, none of that. But go ahead and take a look at these, and it kind of complicates our understanding of sex. Okay, it's traditionally thought that we have male and female. Well, here with these other types of sex-linked chromosomal abnormalities, we see that it's not that simple. Okay, that there are a wide array of different sexes as well. Individuals within that have these different types of chromosomal abnormalities may determine whether or not they're a particular sex. Okay, and that's for them to make that determination. Okay, gene-linked abnormalities. So we have sickle cell anemia and phenylketonuria. Now, I'm going to focus on phenylketonuria. Okay, somebody with sickle cell anemia, you're going to have blood transfusions. You're going to have some medical assistance to be able to live a healthy life. Phenylketonuria is a very interesting disorder in that you can affect change by just abstaining from phenylalanine. Okay, if you have a Coke, maybe you're drinking a Coke right now, look on the ingredients list and you'll find phenylalanine. Okay, it's in many sodas, and you can find it there, and it's in many of our beverages, and you can find it. And individuals that have PKU and they consume phenylalanine will express a phenotype of intellectual disability. Okay, however, if they abstain from phenylalanine, they express a normal phenotype, and their IQ is relatively normal. So this kind of drives home that point that there is an interaction between the environment and the genes. Okay, and a very real and meaningful and purposeful interaction. So if you go back to Urie Bronfenbrenner's work, it doesn't really make sense to separate nature from nurture. They're interactional mechanisms. The genes don't work without an environment. Okay, and the genes are there for the purpose of almost like a heuristic, almost like a pre-programmed heuristic or algorithm for dealing with the environment that may be there. Okay, it's a shortcut route to dealing with typical environments that this individual may need to survive within. Okay, this is an example of mitosis. Please take note of the different of the cellular division and the phases in cellular division. This is an example of meiosis. The difference here is we're making gametes. We're making sex cells for reproduction. Male gametes are called sperm. Sperm have a flagella so that they're mobile. Okay, there's a lot of interesting things that go on with sperm that I don't want to get into too much detail about. But I will talk about one difference between animal species. Okay, there are differences in testicular size. Okay, species that have a lot of breeding competition in that many males may have access to one female. Okay, in those species, you see males with relatively large testicles. Okay, the reason for that is there's a lot of sperm washing that takes place. So male's sperm are literally in competition with another male's sperm. Okay? You see this in chimpanzee. They have relatively large testicles. There's a lot of competition for access to mates. Many individuals will get access to that mate, and there's a lot of sperm washing that takes place. So they have very large testicles with relatively large ejaculates. Now, in species where there's more monogamy, you see smaller testicular size. For example, gibbons. Gibbons are a type of primate that typically their type of ape that typically have monogamous relationships. Okay, they don't have very large testicles. There's not a whole lot of sperm washing and sperm competition. Now, in humans, we're in between. Okay, we don't have as much sperm washing and competition as chimpanzee, but we don't have truly monogamous relationships like the gibbons. Okay, and you can find some other interesting facts about sperm. One other interesting thing that I'll just touch base on, because the production rate of sperm is so high, there are opportunities for epigenetic influences on the sperm, and people are looking into that. Like, okay, can nutrition change the quality of sperm within this individual, and how quickly, if that can happen, how quickly can it happen? Females are born with the eggs that they're going to have their entire life. They have many more than they're ever going to need. You can go ahead and take a look at that video and learn a little bit about fertility and what can be done. Some individuals are choosing to preserve their eggs for a later date. This kind of, makes some sense. If you know you need to postpone reproduction or want to postpone reproduction, but you may want to have kids, then this may be something that's important for you. The egg is a true cell. It contains all of the necessary organelles to sustain life, and this is where an individual will get their mitochondria. Okay, germinal period. The start of this is once the sperm and egg fuse, okay, to form a zygote. It rapidly begins to divide, forming a blastocyst, and then finally a trophoblast, and then it will eventually implant in the wall of the uterus. This is its typical course to the uterus. Fertilization is going to occur in the fallopian tube. Sometimes you can have a lot of very serious mistakes made in this process. By chance, you can have an individual that starts to fully form in the fallopian tube. This is a very dangerous scenario for mom, and a very dangerous and non-survivable scenario for the offspring. Okay, so for this process to go right and for us to be out here is really an amazing thing. There are so many things that can go wrong during the reproductive process that it's a wonder any of us are here. Embryonic period, we see that there are already specialized layers of cells. And what I want you to pay attention to is I want you to pay attention to the ectoderm. The ectoderm is the layer of cells that will eventually become the nervous system. They're already scheduled to become the nervous system. They already have the programming to become the nervous system, as well as the skin. Further information about the embryonic period, just some structures that I want you to be familiar with. The amnion is the sac that's going to contain fluid within which that individual is going to develop. Okay, it's a controlled environment for the fetus. The umbilical cord is going to provide nutrients. It's going to be that highway of nutrients for the developing fetus. And the placenta is very important because it creates a barrier between the fetal blood and mom's blood. Okay, and that's important because we don't want everything that mom's taking into her body to get to fetus. Now, there are some substances that don't play by the rules. They ignore this placenta and just travel right across it. And one of those substances that doesn't play by the rules and is a very serious deal is alcohol. Alcohol can can diffuse right across the phospholipid bilayer, right across the cells. It can go right across this placental barrier and affect fetus and its development. The fetal period, the starting is about 2 months. So we're moving into a period where we start seeing organs become active. We're going to start seeing some movement. We're going to start seeing this infant or this fetus at this point not start to understand associations. Its brain is going to become active. There are going to be neurons that are proliferating and coming online, and there's going to be neuronal migration where these neurons migrate to where they're supposed to be, and then there are going to be synaptic connections formed between these neurons. Okay, so they're going to link up, and all of this is happening over the course of 9 months. Okay, teratogens are substances that can impact or negatively influence the development of this individual. And I'm going to let you watch the videos, and I want you to pay particular attention to what radiation and alcohol do to neuronal migration. Okay, does radiation cause the neurons to stop short or migrate too far? Does alcohol cause the neurons to migrate too far or stop short of their desired location in the brain and the nervous system? Excuse me. Other forms of teratogens include prescription medications. We already talked about alcohol, and we already talked about radiation, nicotine, methamphetamine, cocaine, marijuana, heroin. Any drug, including caffeine, has been hypothesized to influence development in some way. Okay, going back to caffeine, even influences the rate at which sperms swim. So kind of skewing that line as to when does development really begin? Is it at conception, or is it beginning in prior generations? I mean, there really are some philosophical and very scientific questions that are asked about gametes and their roles and the effects of the environment on the quality of gametes and the effects of the environment on the fetus. And that's what we're seeing here. It doesn't just have to be drugs. It can be viruses, and illness can influence the fetus. So we talk a little bit about Zika. Zika can have an influence on the development of a fetus, and it's a teratogen. Okay, so here we get to some developmental abnormalities. I'm just going to go ahead and say this. Because of all of the things that can go wrong in the wiring and development of the nervous system and of the body, it really is a wonder that we're here. We could have a neural tube defect where the neural tube isn't within the body. It's not formed properly, and so you get an individual that doesn't survive. You can have issues with the setup of the prosencephalon, and you could end up with cyclopia, where you, for the most part, only have one eye. And a lot of these things are really, it's kind of sad to see. It is very sad to see these things, and you just kind of feel that, man, those are really big mistakes. Those are really big mistakes that chance made, or whatever you believe in. Those are really big mistakes. in the developmental process. And you can go look at some of those images. I warn you though, I didn't include them here because some of them are they're going to evoke some feelings. You can have mistakes in proliferation where you don't have enough neurons that were formed, and so you get an underdeveloped brain. That's microencephaly. Microcephaly is where you have an underdeveloped cranium, so the skull is not big enough. You see both with Zika. Megalencephaly is where you have an overdeveloped brain, and some have proposed that there may be some benefits to this, but we also see some issues with it. Neuronal migration, you can have some issues there, and you can have some issues in the way that the brain wires itself. So all the way from just forming the cells to where the cells need to be to their connectivity, all of that can be affected. And where we see neural circuitry issues, we may see autism, may see ADHD, and some other things. So it is a wonder that we got here and we're even here at all. There are a lot of steps you have to go through before you get the to be one of the lucky ones that get to live out here. All right, prenatal diagnostic tests. We have ultrasound sonography, and I want to back up. I do mean one of the lucky ones. There's a lot of chance involved in these these processes. It's just luck. Just dumb luck, okay? Just a nice roll of the dice, okay? Ultrasound sonography can reveal some things about what's going on prenatally. There are other techniques that are used very conservatively because of their risk to the developing fetus, and so they're very careful about when they use these other techniques to take a look. Here's some more techniques. Your book does a pretty good job of taking a look at these things. There are some ethical concerns about fetal sex determination in that you may have some cultures that value one sex over the other and may end up selecting against a particular sex. And so there are some ethical concerns within the medical community and within culture at large as to whether or not we want that. And so those are questions that I'll leave up to you. I like this article about the ethical considerations for prenatal diagnostics. This was a German perspective, and this individual was a pediatrician. So this individual is looking at it from a different perspective than a gynecologist. Okay, so keep that in mind. And here are some of the ethical questions that were asked. What's the value of life? Culturally, what's the value of life? What's the value of life to the parents? What's the value of life maybe to the future offspring? Okay, these are all questions that need to be asked. What are the future neuropsychological outcomes of this life? Is this life going to have a neural tube? Is there a neural tube defect? Is there going to be extreme levels of impairment for this individual? Is this individual likely to live a healthy life? Could this individual live 40 or 50 years of their life before they're diagnosed with some chronic illness? Is that a valuable life at that point? These are questions that are asked. What are the prenatal preferences for the unborn life? In other words, what do parents want for their offspring? Okay, what do they want? What's their preference? What's best for the future child? Is this future child going to live a life with a relatively subjectively adequate health span? I mean, is this going to be something that this child's going to benefit from? So these are all questions that need to be asked and addressed when utilizing these prenatal diagnostics. De Jong 2011 talks about some advances in prenatal screening. You have this article. Go ahead and read through it and see what you think. Okay, there are a lot of interesting questions that were posed within the article. For example, anticipatory autonomy rights. Huntington's disease. If the individual has certain genetic abnormalities, it's very likely that they're going to develop this disorder later that's going to be very debilitating. Is that okay? Is it something that we should come to consensus about? Is it choice on the parent's part? Are you stripping the individual of some right that they may have later to make a determination? Is it possible that there could be advancements in technology that help that individual survive? And without any false hope here, there is some interesting technologies being used, like CRISPR-Cas9 gene editing techniques that may be able to help with some of these genetic disorders that we know are very much related to genetics. So there's a lot to consider in prenatal screenings as to, what course of action should the individual take. Okay, and all parties should be considered. Mom should be considered. Culture at large should be considered. The future offspring should be considered, and there's a lot of ethical issues that go into making final determinations from these prenatal screenings. The birthing process. There's stages of birth. They're outlined here. One thing is when you get to multiparous parents, when you get to a mother that's already had offspring, sometimes the progression through this birthing process can be expedited. Okay, but on average, this is what is seen. There are various methods for childbirth. A lot of those methods are related to the culture. So there are very interesting discussions and literature out there about cultural differences and preferences in childbirthing methods. So I'll let you go ahead and read through some of those on your own. This is an APGAR. Okay, you can score max. So there are 5 different categories. Maximum score of 2 in each category. Maximum score in the entire APGAR is a 10. Anything from about a 7 to a 10, we're considering that the individual's in pretty good health. Once we get to like 6 and 7, and 5, we start worrying and grow concerned about what this individual needs, probably some medical attention. And anything lower than that, we have a very serious situation. Brazelton's assessment, the neonatal neurobehavioral assessment was designed to test infant reflexes and to look at the very basic behaviors of a neonate, of a newborn. Okay, and that assessment was designed for relatively healthy infants that we don't suspect were exposed to teratogens or didn't have heavy exposure to teratogens. Then the Neonatal Intensive Care Unit Network Neurobehavioral Scale, the NNNS, was developed to assess similar things, infant reflexes, posture, psychological processes such as the ability to habituate to stimuli, and all of that was designed specifically for at-risk infants. And so at the end of the assessment, they also go and look at different features that relate to exposure to specific teratogens. Okay. And you have this Neonatal Intensive Care Unit Network Neurobehavioral Scale. You have the entire article. It's a long read. Please just skim through it. There aren't very many specific questions about what's going on within that assessment. I certainly don't expect you to know how to use it. But we already talked about it a little bit. Appropriate ages are where the gestational age was greater than 30 weeks and less than 48 weeks. So between 30 and 48 weeks. Now, those are appropriate parameters for this assessment, age parameters for this assessment. It is a semi-structured assessment in that there are certain things that are going to stop the assessment or slow the assessment or lead to a different coding of a particular feature. For example, if the infant's not in a particular state, you may delay future assessment, or you may delay proceeding through the assessment. If the individual never gets to a certain state, you're going to make note of it and then continue on. But it's not a and there is some subjective reporting that occurs through that process. And I'll let you watch that video by Dr. Lester down there talking about services for neonates and newborns. Okay, postpartum period is interesting. There is an impact on parents. Sleep deprivation is reported by new parents. They're adjusting their life. This is a significant life event for them. If you've taken general psychology, you know that things such as marriage and even really good things like marriage and really good things like graduating and really good things like getting a new job and a promotion and having a kid, these are very exciting things, but they also lead to changes in your life that can produce stress. So it's very important for the individuals to have coping strategies for dealing with that stress. And having a newborn is very exciting. It's a good thing, but it can be very stressful. Hormone levels may change. Oxytocin changes in mom at birth. A lot of this is to form that bond between mom and infant, which is a very appropriate bond at this point. Oxytocin promotes within group bonding, so mom and infant need to be a group. Mom needs to want to watch out for this offspring, and mom needs to be able to very readily identify offspring and those feelings from somebody that may harm offspring. And so that's what oxytocin does. It creates that within group, in group bonding, and helps readily identify out group members and respond differently to them. And so this is appropriate for mom at this period of time. Moms can have postpartum depression. One of the things that you may look at is oxytocin levels in mom to kind of get an idea of what's going on. There are other things and behavioral assessments and cognitive assessments that can assess if mom may be at risk for postpartum depression. If so, seek help. Don't go it alone. Okay, a responsive mother is very important for the future development of that infant, and mom needs help herself. She's going to need that assistance to recover from this, and a lot has changed in mom over the course of those 9 months and through the birthing process. And if mom's feeling, symptoms of depression, it's okay for mom to seek help, and mom should want to do that for herself as well. Fathers may feel as though they've been replaced by the infant. Fathers have a very large investment in heterosexual couples. Fathers have a very large investment in mom, and that's where their relationship is, and they, dads can be very good with offspring, but it takes time for moms and dads to get, good with offspring. This is a new individual, and they don't really know how to relate to it yet. It's not at all that instinctual. There's a lot of learning that occurs on the job here, but fathers may feel replaced, and so that may lead to some depression or in them as well. in other forms of relationships, it may be that certain roles are adopted and that certain people feel various ways depending upon the dynamics of their specific relationship. Okay, the bonding hypothesis says that infants need to bond with their parents very shortly after as quickly as possible following birth. Okay, this may help mom, may trigger certain hormone releases in mom, may provide infant with very quick access to nutrients and antibodies that they need that may help them survive. Okay, and so these are very, it may not be a critical period, it may be a sensitive period of development, but bonding is relatively important. Okay, here's some references, and I apologize for that top reference. It's kind of odd that it got pushed up there. I didn't notice that before, but that's Darwin's article that you can find from on York U's website. So I hope you enjoyed this particular lecture. Remember and keep in mind that biology is very important and we need to understand it. We also want to understand the context within which these neural and biological correlates relate to behavior and mental processes. So I hope you enjoyed the lecture yet again, and if you have any questions, feel free to email me.